Howdy, I'm Professor Carafit, and today we're going to continue the cell membrane lab. You did the first cell membrane lab in person this semester, where you looked at spectrophotometers and you made a standard curve of beta-cyanine absorbance. The basic research question that we would address this week is, what is the effect of ethanol on the beetroot tonoplast membrane? And why might we care about this? Remember, we're not really all that interested in beetroots for their own sake. We're interested in them because they have membranes just like we have, and the tonoplast, which is the membrane around the central vacuole, contains, well, within the vacuole is beta-cyanine, which is red, so we can see when it's damaged because red stuff will leak out. So when we think about ethanol in membranes, why might you care about how ethanol damages membranes? Well, think about what's in that hand sanitizer you're rubbing on your hands every day. Why are you doing that? What effect is it having on the membranes of the external surface of coronavirus, for example, or bacteria on your hands? Why does it work? What's it doing? So a few questions that you would need to be able to address in order to really understand this project is, what is the function of a cell membrane? You can read about that in your textbook, but it is the ultimate barrier between the cell and the outside world. It's what allows things in and out of a cell. It's what contains the organelles and cytoplasm within the cell. What is the structure of a cell membrane? What's it made of? And what kind of things might damage it? So just so you know, in case you haven't had this in lecture yet, cell membranes are composed of phospholipids. So we have a phosphate group up here, which has a charge, so it's polar, and we have fatty acid tails, which are nonpolar. So part of that molecule can interact with water. It's hydrophilic, the polar part, and the hydrophobic part is repelled by water. At the same time, ethanol is amphipathic, meaning it has a polar part and a nonpolar part. So what could be happening is that ethanol might be dissolving phospholipids, so the polar part might be interacting with the phosphate group, so the nonpolar part could be interacting with the fatty acid tails. Or ethanol might be denaturing the proteins in the cell membrane. So cell membranes are composed of a phospholipid bilayer as well as proteins like we see here. Here's a protein, here's another one, that these transmembrane proteins that cross the membrane could be denatured. That means their shape changes and they become basically a hole where beta-cyanine could leak out. So that could be something that's happening here. Other things that could damage a membrane would be heat, if we were going to heat these up and damage the membrane that way, because as you add energy, molecules move and they spread out and proteins denature and things like that. So just remember the protein where the beta-cyanine, I'm sorry, the location of the beta-cyanine is within the central vacuole, this organelle right here, and this membrane is called the tonoplast. And so when we add ethanol to beet tissue, we're damaging both the plasma membrane and the tonoplast membrane. So beta-cyanine has to leak out into the test tube for us to see the damage. So within the central vacuole, that's where beta-cyanine is found. And you've done a whole lab on this already, making a standard curve, which you're going to need today. So dig that standard curve out. So basically, what we would do now is we would have to think of a hypothesis, and I'm going to provide you with an example of one today. Write a predictive hypothesis. So if, and then. And so an example might be something like, if ethanol dissolves lipids or denatures proteins and beet tissue is incubated in a gradient of ethanol concentrations, then there will be little damage at low concentrations, but damage will begin to increase as ethanol concentration increases. That hypothesis could be refined and it has a typo or two in it, but still, it gets the point across. So if ethanol damages membranes, one way or another, right? We're trying to figure out how, like if it dissolves lipids, and we put pieces of beets in lots of different concentrations, then we're going to see more damage at higher concentrations. And actually, I didn't work it into this hypothesis, but we know that beets can make their own ethanol in anaerobic conditions, meaning when there's not a lot of oxygen around. So we think beets are probably resistant to low concentrations of ethanol because they have evolved to tolerate low concentrations of ethanol. But that's something you could work into your hypothesis as well. So I'll ask you, what are the independent variables today? You should be able to answer that. And what would a control be? Once again, if you're testing various concentrations of ethanol, what would the control be? Well, the basic control would be 0% ethanol. That would be a negative control, no ethanol present. You could also test 100% ethanol to ensure that ethanol really can damage cell membranes. But ultimately, what we would do in lab typically is have 0% for our basic control to see if water damages cell membranes, then what's the point of testing ethanol at that point? So the way we would do this is we would cut up a beet. We would take what we call a cork borer, which is just a hollow tube here, a cork borer, and we would stab it into the beet, and we would get a cylinder like this, which we would then cut up in pieces, into discs that are 5 millimeters wide. Then we would rinse those discs off in the sink because we don't want to measure beta-cyanin leakage from a knife cut on a beet. We want to measure alcohol-induced damage, right? So we rinse off the beet, and then we place it in various concentrations of ethanol. So you would make dilutions of alcohol, and you would put the beets in there for 5 minutes, and then you would take the alcohol out with a pipette and see what its absorbance was using a spectrophotometer. So here we can see in this test tube on the left is water. It's just a very light pink, and over here is 95% ethanol, which you can see is much pinker, but that's qualitative data. We need to quantify it with the spec, which you've already done, so you know how that was done. So basically what we would do is we would blank the spec. This time we would blank it with water and alcohol, a 50-50 mix, because the concentrations you were making were water and alcohol, and we're not interested in the absorbance of water and alcohol. We want to know about the absorbance of beta-cyanin. So we blank the spec. Typically with water and alcohol, though water and alcohol have about the same absorbance, so you could really blank with just water probably. And then we would collect a bunch of data. Then we would use our standard curves from the previous week to convert that data, that absorbance data, into concentration. And the best way to do that is to use the equation of the line. Y equals mx plus b. And because you have a standard curve, you know what m is and you know what b is, because you can go to insert best fit line, show equation of the line in Excel, like in the videos I made for you. You can see how that's done. So this is an example of a standard curve, and what we see here is if I have a question, if something had at 535 had an absorbance of 0.398, what was the beta-cyanin concentration? So how could you solve that? Well, absorbance is the y-axis, right? And we know that it's 0.398, so it's about right there, right? The best way to solve this is that, since this is the y-axis, we put 0.398 in this equation for y, and then we solve for x. So this is the x-axis down here. X is beta-cyanin concentration. So we simply set up the equation, x equals y minus b over m, and we solve the equation, and we get our answer. X is 6.99 micromolar concentration. So if you set up your equation of the line properly, you can plug these in and answer them very rapidly and convert them. You can't just convert your averages. You have to convert all your data to micromolar and then do your averages and then make a graph based on your data from this lab. And the data is provided on the lab website. Here's an example of what a graph might look like, okay? I just want to show you this. So you have ethanol concentration on one axis and beta-cyanin concentration on the other. Please use proper units. So use the micro symbol for micromolar. You have to go to symbol insert in Word and hunt for it and find it. And once you use it, then it will become easy to find in the future because it will be a frequently used item on your computer. These brackets around these words mean concentration, so you don't have to type the word concentration. And here's an example of the graph. Now, I don't have the caption here written for you here, and I don't have an example caption, but you can see an example of data from a previous semester. And notice we have standard error bars here, and they're not all the same size, which is a good sign that they're probably correct. Notice in this case, these data points aren't that different from one another. And what we use these error bars for in this case, we say if these error bars overlap in space, see how these error bars take up some of the same regions of the y-axis, then we can say these different data points, so the averages are different. They're likely not significantly different because the error bars overlap. However, that's not a guarantee. So that's why we use the word likely. Because if you wanted to know if they are significantly different, we'd have to run an ANOVA analysis or something like that, a more robust statistical analysis to see if they were significantly different or not. I can show you how to do that if anyone's interested. We have a pretty easy ANOVA analysis we can run, but we don't need to for today's lab. But we can see down here, these are all about the same. This one, we see an increase. And by the way, if all the error bars kind of overlap and you're not sure if anything's different, that's okay. You can talk about the trends of the data. The trends of the data are this line that you see here. That's not a best fit line. That's just a connect the dots line. The trends are the overall shape of the data. So in this case, the data does not change much, and then it begins to increase until we reach 50% ethanol, which was tested here. We can see that it increased that entire time. So that's an example. Now, some data, this might level off then, and if that happened, it would be called a plateau. So you need to be able to interpret your data and quantify the change. So if I were describing this change, I would say there was very little change from 0 to 30%, although I could quantify a slight change from here to here. And then I could quantify from 30 to 50% ethanol. What percent change was that in my figure caption? Check with your instructor on the assignments for this week, but one of them is going to be completing an assignment based on the data that we provide you and using your standard curve. The other assignment is going to involve reading this paper, which is an example of a scientific paper, because it follows a similar format to our lab reports, and soon we will be writing a lab report. Stay tuned to your lab instructor for further details. But you're going to have an assignment based on this, so read this paper carefully, see how it's written, how the wording is put together, see what goes into each section, and just think about the style of writing, how it's different from your English courses, for example, how concise and precise the language is, because you're going to try to sort of replicate that in your own writing this semester.